Welcome to Life Biology. I'm Dr. Joel Graff, and today we're going to be talking about cell cycle regulation and cancer. This is a lecture I gave earlier today for my Biology 101 class. It's called Discover Biology. So, jumping into cell cycle regulation, the first thing we did was we discussed that we did a review of the cell cycle, which was discussed in our previous class. Some things to notice here are that you start with one cell, you go around the cell cycle, and you end up with two cells at the end. And then each of those new cells can go through the cell cycle again. So you can go from one cell to two cells to 4 cells to 8 cells. Each time you go around here, you have a doubling of your cells. Okay. The next thing to know is that this first arrow from here down to the arrowhead here is a phase called G1, and that is a phase where the cell is doing some growing. The next phase is called S phase, and the S stands for synthesis, and in this phase the DNA, the genome chromosomes are going to get copied. Then we're going to move on to G2, which is another gap phase or growth phase. And then the final stage of the cell cycle is labeled M here, but that's short for mitosis. Let me pull up so M equals mitosis. In the near future, we're going to be talking about meiosis, spelled a little different, and also involves quite a few different steps. So anyway, we're talking about mitosis here, and this is just your typical one cell goes to two cell. Another part of the review we talked about was that interphase consists of G1, S, and G2. So those three different phases where we kind of separated out can be lumped together if that happens a lot when you want to focus on mitosis, for example. There is a G0 stage, and that's when a cell exits the cell cycle. You have some cells like nerve cells, muscle cells, adipose cells that aren't known for replicating. And so what happens is that once a nerve cell is established, it exits the cell cycle and just hangs out for the rest of your life. Hopefully. So anyway, those are the stages of the cell cycle, and last time we weren't able to get too caught up in the phases of mitosis, and so let's go through mitosis real quickly here. So I took an image from Shutterstock, hopefully that's legal, and I threw it up on the board, and then I added some information to it. But let's just go look at the Shutterstock stuff first. So it's saying interphase, your cell looks like this. Prophase, metaphase, anaphase, and telophase. Okay? So those prophase, metaphase, anaphase, and telophase, those are the small green arrows we have up here. I drew 5 arrows because occasionally prophase is also shown to be separated into pre-prophase and prophase. Okay. The first thing we saw over here was the DNA is loosely organized, and in the rest of the images the DNA is condensed so that we can see those nice X shapes of the chromosomes. They're sister chromatids. It started out as one chromosome. It was copied during S phase, and the two copies are held together near the middle of the X, which is some centromere sequence. And then we have, again, metaphase, anaphase, and telophase. The next thing to look at is the centriole, and I didn't write that very well, so let me try to write it a little bit clearer. Centriole, or centrosomes, one of those. Anyway, we go from having one copy to 2 copies. Back in interphase, the centriole or centromere centrosome. Too many to keep track of. It sends out microtubules from near the nucleus of the cell to the outside of the cell, the plasma membrane. So things, if they get into the cell, they could take a ride on a microtubule to get into close to the nucleus. Or if things are made in the nucleus, they can take a ride on the microtubules to head out to the surface of the cell. We'll talk more about that with the endomembrane system later in the semester. Okay, so the DNA was loosely organized, and then it's over here, it is condensed. Another thing that happens as you go from interphase to prophase is that you lose the nuclear membrane. So the DNA was within a nucleus here, that nucleus is gone. Also, when those centrioles divided, they moved to opposite sides of the cell. So this will be, we're going to call this the North Pole and the South Pole. Again, you've got the North Pole here and the South Pole here. The difference between prophase and metaphase is that the microtubules that disintegrated back at this stage reform, but this time they go and they grab the centromere sequence. It's probably that these are centrioles and these are centromeres, but you can look it up on Google. Anyway, you pull those, the microtubules will connect to each of those X's. Each of those X's is connected to the centrosome, or centromere, centriole. Should probably know the name of that. But they're at the opposite poles of the cells, and if both of the if the X's are being tugged on, sister chromatids are being tugged on from both poles, then it makes the chromosomes line up down kind of the equator of the cell or the middle of the cell. And to remember which phase is associated with the chromosomes at the middle of the cell, metaphase starts with M, middle starts with M. So that's how you can remember that one. Anaphase, those microtubules start pulling on the chromosomes, and your sister chromatids break in half so that you have a chromosome of each kind ending up at each pole of the cell. And then the cell divides during telophase, and it's not written here, but there's cytokinesis. And the nuclear membrane reforms, and the cell has elongated here and starts to show a slight narrowing in the center, and it gets narrower and narrower until these separate into two separate cells. Over here we had microtubules as the cytoskeleton features that were helping out with pulling on the chromosomes, and over here this is a different type of cytoskeletal structure. It's made of actin filaments. So we've got some actin filaments going there, and they're going to restrict and get smaller and smaller and pull past each other until the cells divide into two. Okay, after the cells completely divide, the DNA that had been not condensed, then it was condensed through here, will uncondense again, if uncondensed is a word. Okay, so that's mitosis in a nutshell. So we're back at this cell cycle. We have G1, S, G2, and then the several steps of mitosis. I want to point out that there's 3 places where I drew in a stop sign. At least that's what they're supposed to be. And these are called checkpoints. Some textbooks have a checkpoint in the S phase, but we're going to ignore that. So we have a stop sign here at the end of the G1 phase, so we're going to call it the G1 checkpoint. We have a stop sign towards the end of the G2 phase, so we're going to call it the G2 checkpoint. Checkpoint. I just realized that my ugly mug is in the way, so hope you don't mind, but I'm going to bow out now so I quit covering up the material. What was on the on the previous slide. There's some notes down here, but I basically talked through it. Okay, back to those checkpoints. And then there's a third checkpoint right in the middle of mitosis, the stage that starts with an M, because we go prophase, metaphase, anaphase, telophase. There's our pre-prophase, if you want to have 5 steps. And this checkpoint right here at the M phase has its own purpose. So that's called the metaphase to anaphase checkpoint, something like that. Anyway, at the G1 checkpoint, a couple things need to happen. You need to have some growth signal hitting the cell during G1 to get the cell over to this point anyway, and then you also need, the cell needs to check for DNA damage. If there is DNA damage, it'll stop and it'll try to fix the damage of the cell. If the damage can't be fixed, the cell will decide to go through a process of programmed cell death called apoptosis. We'll come back to that a little bit later. But they stop here, check for DNA damage. If it can be fixed, we move on. We go through S phase and copy the DNA, and then we get to the end of the second gap phase or growth phase, and there's another checkpoint. And that's because a lot of things happen, like the nuclear membrane breaks down and all those sorts of things, to get us through mitosis, all these steps of mitosis. So we have a checkpoint again. And just like we stopped and checked for DNA damage at G1, we're going to check for DNA damage again, because remember, it's been copied during S phase. So if the DNA looks fine and the cell decides to go on into mitosis, the cell will stop once again at that midpoint metaphase of mitosis, and we want to ensure that each pole, that is each new cell, gets the right complement of DNA. What that means is that when these microtubules are extending out and grabbing onto these sister chromatids, you want to make sure the cell does a little bit of a tug-of-war thing with all of these different sister chromatids, and it makes sure that the sister chromatids are being grabbed at both ends, because you want to make sure you have that true for every single chromosome. In your human cell, that would be 46 chromosomes lined up here. You want to make sure they're all good before you pull apart, because if you did not have the spindle microtubule was not attached here, then you wouldn't pull this yellow chromosome, the shorter yellow chromosome, up. It would end up pulling both sister chromatids down here, so that then you ended up with 5 chromosomes in this cell and 3 chromosomes in that cell. We don't want that to happen. Okay, so these checkpoints, there are some proteins that are going to be involved in saying you should go through the stop sign, and there's going to be some proteins making sure that you stop at the stop signs. And so that brings up the topic of oncogenes and tumor suppressors. Oncogenes are there, and if they give the okay, it helps the cell decide to go through the checkpoint. One problem is if an oncogene is mutated so that it's always on, it could say go all the time and push the cell through the cell cycle. Oncogenes aren't the only players involved. You have tumor suppressors, like we said. They are involved at stopping and checking the cells out at each of those checkpoints. However, you can get mutations to tumor suppressors as well. They could lose ability to stop at checkpoints if this happens, so the cells could lose the ability. One thing about oncogenes, you just need one of your 2 versions of oncogene to get mutated here. You get an oncogene from your mother and an oncogene from your father. If just one of those gets mutated, you're going to go through the cell cycle. Tumor suppressors, you've got a tumor suppressor from your mother and a tumor suppressor from your father, and if one of those tumor suppressors becomes one of those tumor suppressors becomes mutated, you're okay, but if both of them become mutated, then you've got a problem. Okay, so we're dabbling into the area of cancer at this point, and so one of the concepts that I really like is this concept of hallmarks of cancer. In 2001, there was a Cell review called The Hallmarks of Cancer, and the two authors of this review essentially provided a framework for thinking about cancer. I don't know. Got some extra lines there. We'll try to ignore them. Anyway, the idea here is that we put different categories of cancer biology into separate buckets, and they came up, originally they had 6 different hallmarks of cancer. Later on, 10 years later, they expanded the number of hallmarks to 8. They added some other stuff too, but we won't get into that. But what these hallmarks of cancer is meant to show is that cancer is a multi-step process. So each of these 8 hallmarks are things that can happen to the cell to push it in the direction of being a malignant cancer. However, the cell needs to accumulate multiple ones of these in order to be a successful malignant cancer. So over the course of your life, these hallmarks might start to be acquired by your different cells, and if enough of them combine together, you can have the cell turn into a malignant cancer, particularly if it gets to this point here. But I'll talk about that in a little bit. The other thing that this hallmarks of cancer is important for is it shows that there are different roads to cancer. Just because someone gets a mutation in a tumor suppressor and then later gets a mutation in genes involved in cell death doesn't mean that someone else couldn't get a cancer that has a mutation involved in cellular, maybe sustaining proliferative signals and also avoiding immune destruction. And so the order in which a cell would collect these hallmarks of cancer is going to differ from one person to the next as far as their cancers are concerned. So let's talk through each of those hallmarks for a little bit. First thing I need to mention is that I have an example here that needs to end up here, so I'm going to skip it when I talk through these. So sustaining proliferative signaling, this is when you can, it's supposed to be oncogenes. Let me get a are always saying go. That could happen, and you'd sustain proliferative signaling. Also, growth factors could be made when they weren't supposed to, or a nearby cell is going out of control and it's making some growth factors. I had initially put tumor suppressor, this is TS for tumor suppressors, forgetting to say stop, but that actually fits better into this evading growth suppressors category. Next, we've got avoiding immune destruction, and this is usually the immune system is thought to attack non-self, but what's interesting is that in the recent years, recent decades I suppose I should say, we've come to realize that the immune system isn't just going to attack things that don't look like your body, but the immune system could also attack altered self, which is when your body cells turn cancerous, they might have some features different about them that the immune system could detect. And so if the immune system is typically wanting to kill off cancer cells, and it's quite possible cancers started rising in people all the time, but then the immune system takes care of it and we never even know about it. One thing that can happen is a tumor could suppress the system locally, the immune system locally. So your rest of your body might be doing fine as far as fighting off viruses and bacteria or fixing wounds or whatever it is the immune system's going to do, but there within the tumor that's starting to grow, the immune system is suppressed. Moving on, enabling replicative immortality. So chromosomes will shorten each time they replicate, and in theory there's a finite number of replications that can happen. At the ends of your chromosomes are these structures called telomeres, and so they are the sequences that shorten. It's a length of repetitive DNA sequence. In cancer, you can have an enzyme called telomerase, become active, and it's the enzyme that knows how to lengthen your telomeres. So the cell had a nice system of counting how many times it replicated, but then that system went awry when the measuring tape it was using got extended again and it lost track of replications. Deregulating cellular energetics. So we've just finished talking about metabolism. We went through cellular respiration, and we know that there is aerobic and anaerobic respiration. Aerobic respiration, you get a lot of ATP, over 30 ATP typically per glucose molecule that's broken down. So usually cells will choose the aerobic respiration if possible, if oxygen's around. If oxygen's not around, cells do anaerobic respiration, and the problem there is that you only get 2 ATP per glucose, and then you get some waste products. What happens with cancer is they don't really care about the rest of the body. Those cells in the cancer tumor don't really care, and they can change their metabolism to take up the Warburg effect, and this happens in cancer. Cells will use aerobic respiration. I should say anaerobic. Anaerobic respiration, and they only get 2 ATP per glucose even when oxygen is present. Okay. Also, it creates lactic acid, lactate, and that actually promotes tumors. So not only is the cancer using up all your energy for no real good reason, it's also producing this byproduct of anaerobic respiration that helps promote the tumor. Resisting cell death. Okay, we've got the symbol here is a cross. If DNA damage can't be fixed, cells should die. So we talked about there's two out of the 3 checkpoints were to check to see if there's any DNA damage. So for example, if the cell does have DNA damage, it tries to fix the DNA damage, but if it's unable to get it fixed, then that one cell should die because it's only one cell out of tens of thousands of, 10 million or 10 trillion cells in your body. Yes, 10 trillion cells is in the body. So if one messes up bad, it's okay to kill it off to let the other 10 trillion minus 1 cells live. Recently I heard the number being 31 trillion. I don't know the exact count, but it's in the tens of trillions at any rate. Programmed cell death is what the cell decides to do. So an example of programmed cell death is called apoptosis, and that means to fall alone. Ptosis is to fall, apo is alone. Okay. Next we've got inducing angiogenesis. Angiogenesis is the fancy word that means growing new blood vessels. And in the case of angiogenesis, tumor cells, in the case of a tumor, your tumor cells grow quickly, but if it's just the regular tissue cells dividing, then there's not going to be any blood vessels to bring in oxygen or nutrients or to take away waste products from the tumor cells that are growing quickly. And so tumors need to be really active in promoting the growth of new blood vessels. So that's what's going on with inducing. That means turning on or upregulating or causing the start of growing new blood vessels. And then finally we have activating invasion and metastasis. So sometimes if someone gets a tumor and the doctor biopsies it, they're going to check and they're going to decide if the tumor is malignant or benign. You want benign, not evil. But what it means if it's malignant is that it can move out of one tissue and into others. However, some of the tumor cells stay behind. So what this means is that as your tumor is growing in one tissue, some of the cells flake off. They can get in blood vessels or lymph vessels or other vessels and get carried to another part of your body. So oftentimes, if you have a tumor removed because it's showing signs of being malignant, the doctors will not only remove the main tumor, but then they'll also remove some lymph nodes that are in the area of the tumor, so that if some of the cancer's cells dropped off and went down a lymph vessel and got caught up in the lymph nodes, by taking those lymph nodes out, you're going to hopefully get rid of some metastatic cancer cells. The body has a lot of lymph nodes, and so losing a few lymph nodes with the hope of avoiding the spread of malignant cancer is well worth it. So that's why you'll hear about doctors doing that to people. Okay, so that's the end of the discussion today. So we went over cell cycle checkpoints. We talked about what decisions are made at each of those checkpoints. We talked about the steps of mitosis. When we come back next time, we're going to talk about a different sort of cell division process called meiosis, and so we'll do some comparing and contrasting of those. And then we also talked about cancer, oncogenes, tumor suppressors, and how they fit into the hallmarks of cancer, and as well as the other hallmarks of cancer. Okay, well, that's what we went over today in Biology 101.